Hello everyone, my name is Guzar Pallavi Shivaji. My seminar topic is mu transposition controlling elements in TnA and Tn10. Before moving to topic, firstly we see the basic concept. What is transposons? So it is this term is coined by Hedges and Jacobs in 1974. And transposons are the mobile discrete sequence or DNA sequence in a genome are able to transport themselves from one location to other location within the genome. So it is a DNA sequence in a genome and have the ability to move themselves from one location to other location within the same genome. And these element which having the ability to move themselves from one place to other place are also called as the jumping genes, as well as they also called as the transposable genetic elements. तर ट्रान्सपोझोन्स काय आहेत का हे डीएनएचे सिक्वेन्स आहेत ज्यांच्यामध्ये कॅपॅसिटी आहे का ते स्वतःला मूव करू शकतात एका जागेवरून दुसऱ्या जागेवरती आणि त्यांनाच आपण जम्पिंग जीन्स असं पण म्हणतो. त्याचबरोबर त्यांना आपण ट्रान्सपोजेबल जेनेटिक एलिमेंट्स असं पण म्हणतो. टाइप्स ऑफ ट्रान्सपोझोन्स. आता जे ट्रान्सपोझोन्स आहेत त्यांचे मूव होण्याचे दोन टाइप्स आहेत. तर ते कोणत्या प्रकारे मूव होतात एका प्लेसवरून दुसऱ्या प्लेसवरती त्याचे दोन टाइप्स आहेत. फर्स्ट इज अ नॉन रेप्लिकेटिव्ह ट्रान्सपोझोन्स अँड सेकंड इज द रेप्लिकेटिव्ह ट्रान्सपोझोन्स. सो फर्स्टली वी डिस्कस दी नॉन रेप्लिकेटिव्ह ट्रान्सपोझोन्स. In this case, the transposable element moves from one side into the other. Since there is no duplication of the transposons, it is lost from the original site. In this case, the transposons move from one place to other place without duplication. That means there is no formation of their copy. These transposons is directly transferred from original place to the other place, and that therefore there is a loss of the transposon from the original place. So composite transposons Tn5 and Tn10 occur according to this mechanism. So the Tn5 and Tn10 group of transposons are work in the same concept or occur its transposition also occur in the same mechanism. So with the help of diagram we see this. Let us consider this is a gene and here is a sequence of the transposons, and here is a transposable element. This element cut from the gene and then they move towards the other gene and then they attach to this other gene. But when they attach to this other gene, there is a loss of this gene, this sequence from the original gene. So this is called as the cut paste mechanism. So non-replicative transposons are occur or they work on the cut and paste mechanism. So next is the replicative transposons. The transposons is duplicated during the process of transposition. One copy of the Tn element remains at the original site while the duplicated copy is inserted at the new site. Such a type of transposition is a characteristics of TnA group of transposons. That means the TnA group of transposons are work in according to this mechanism. That means replicative transposons. So what is replicative transposons? In replicative transposons there is a formation of the two copies of the transposons. That means first is the original transposon and second is the copy of the transposons, and this copy of transposon then transferred to the another genome. So this is the one gene and this is the transposons. Okay, this is transposons and this is the another gene, and when this original gene makes their own copies, that means another transposons, and these transposons then transfer to the other gene and then attach with the other gene. So in this case there is a one transposon at the original site and its copy is placed on the other gene. So these replicative transposons are occur on the copy and paste mechanism. Non-replicative transposons work on the cut and paste mechanism. That means when they cut and then transfer to the next genome or other gene, they completely remove from the original gene. But in this case, the original gene make the copies of themselves, and these copy then transfer to the other gene. So this is the cut and copy and paste mechanism. Next is main topic, that is mu transposition controlling elements in TnA and Tn10. So mu is a bacteriophage and they are called as a transposons because they also acts as a transposons. It is a very large and is regarded as a giant mutator Tn element. So bacteriophage, mu is a bacteriophage and it is a mutator Tn element. So hence they are called as the transposons. These transposons have the ability to insert into E. coli chromosome at a different non-homologous position. The enzyme mu transposase cut both the ends of the transposons from the donor molecule and recipient, that is the target molecule. Mu utilizes both the replicative and non-replicative pathways of the transposition. So here replicative and non-replicative pathway are used in the mu transposition. That's for that purpose we already discussed what is replicative and non-replicative pathway. Its integration into the host genome occurs through the non-replicative process, while the multiplication of mu occurs through the replicative mechanism. So in the mu bacteriophage, it is a transposons. When these transposons are then inserted into the E. coli or the plasmid of the bacteria, at that time its integration into the host genome occurs through the non-replicative processes, while its multiplication occurs through the replicative mechanism. The enzyme mu transposase recognizes the terminal sequences and makes staggered cleavage at the end of the mu and also at the target site in the chromosomal DNA. Let us consider this is the two genes. These are the two genes, and mu transposase, when the mu transposase recognize that there is a presence of the mu transposons on a one genome, it makes the cleavage at the one end of the that transposons. Suppose this make cleavage at this end of the transposons. At the same time, it make the cleavage on the one end of the other genome. Suppose it make the cleavage at this point on the other genome. Then one cut end of the mu joins with the cut end of the target site of the recipient chromosome, producing a cross-shaped structure. Now the cut end of the transposons then join to the cut end of the other genome, and the cut end of the other genome then joins to the cut end of the transposons, and there is formation of the cross shaped structure. Here I draw this structure. This first genome, this is second genome, and when transposons is cut from this region, the transposons become or like this, and this other genome, then this other genome like this, and there is a formation of the cross structure like the crossing over, and there is a recombination or the recombination is take place, or the transfer of the genetic material is take place. And here in the next step there is formation after the recombination or exchanging its genetic material, there is a formation of the new strand is take place. Let us understand with the help of diagram. This is donor molecule and this is recipient molecule, and here is a transposomes present on the donor molecule. So then there is a formation of or the cut end of the transposomes join together or join to the cut end of the target DNA, and then there is formation of the crossing over like structure, and after crossing over there is a transfer of the genetic material also take place in the transposomes and the target sequence. Now the next process is followed by a two method. First is a non-replicative and another is a replicative. So in some cases it is followed by a replicative mechanism, and in some cases it is followed by the non-replicative mechanism. So in case of non-replicative mechanism, these transposomes are then transferred into the or on the recipient molecule and or inserted into the target DNA of the recipient molecule, and there is a loss of transposomes on the donor molecule, okay, donor sequence or the donor strand. Then that means it is followed by the cut and paste mechanism, okay. In case of replicative mechanism, the replication from the 2-3 ends to the produce the two copies of the transposomes in the single co-integrate molecule is take place. So here this is a co-integrate molecule. Co-integrate molecule means it contains the target as well as the donor molecule sequence as well, that means the target sequence as well as the donor sequence in the gene or the DNA of the plasmid or the bacteria. So after that, in the next stage there is a formation of or there is a separation of the target molecule as well as the donor molecule is take place, and in both the molecule there is a presence of the transposomes. This is a transposon. So both the target molecule as well as the donor molecule have a transposomes. So this is called as the replicative mechanism. That means the transposomes is present on the donor molecule as well as it copied on the target molecule or the recipient molecule also, and this is called as the copy paste mechanism. Okay. Next is a cleavage in the donor molecule near the transposon causes the insertion of the transposon in the target DNA. The single-stranded region formed by the staggered cleavage is filled by the repair synthesis. This is the non-replicative mechanism in which donor DNA suffer a double-stranded break and loses the mu transposons. So here the process of the non-replicative mechanism is given, and we already discussed non-replicative as well as the replicative process with the help of the diagram. So next is the TnA transposons, and the next is the Tn10 transposons. Firstly, we discuss about the TnA transposons. Transposons of the TnA group such as Tn3, Tn100, etc. are the large transposable element containing about 5,000 base pairs. So this is the large group of the transposable elements, and it contains Tn3, Tn100 elements and more elements in this group. They carry genes required for the transposition along with the gene for resistance to antibiotics such as ampicillin. The enzyme transposase coded by the gene TnPA and resolvase coded by the gene TnPR are involved in the transposition of TnA transposons. So here these transposons, that means the TnA group transposons, also carry the genes that is for the resistance to antibiotics such as the ampicillin, that and the enzymes like the transposase. These enzymes are coded by the gene TnPA and resolvase coded by the gene TnPR. Here transposase, we have already seen the transposase used for cutting the cutting the mu bacteriophages or mu transposons from its end on the donor molecule, but resolvase is used, or its use is for the separation of the integrate. This is firstly, this is the integrate in which have the both the which contain both the donor sequence as well as the recipient sequence in this plasmid. So for the separation of the donor as well as the recipient molecule or the target molecule, the enzyme resolvase is used, and it coded by the gene TnPR and involved in the transposition of TnA transposons. Next, the transposase bind to a sequences of 25 base pairs located within the 38 base pair inverted terminal repeats. These enzymes make a staggered cleavage of 5 base pair at the target DNA. The transposase binds to a sequence of 25 base pair located within the 38 base pair inverted terminal repeat. That means this transposase enzyme bind to the base pair of the donor molecule or the donor genome at the 25 bases of the out of the 38 bases of the inverted terminal sequences, and then they cleave the sequence from the 5 base pair at the on the target genome or the DNA. This product of gene TnPR function as a repressor of the TnPA as well as a TnPR itself. The repression of gene TnPR or the mutation in the gene TnPR result in an increased synthesis of transposase. If there is a mutation in the TnPR gene used for the resolvase or resolution of the integrate, but if there is a mutation in this TnPR gene, then there is an increase in the synthesis of the transposase, and this transposase make a cut on the donor gene or the DNA sequence at both the ends of the transposons. This lead to the higher frequency of the transposition, that it causes the formation of the more transposons or the more copies and the higher frequency of the transposition. The enzyme resolvase is involved in site-specific recombination between the two transposons present at the direct repeat in a co-integrated molecule. Next is Tn10. Tn10 is a composite transposon carrying the gene for a tetracycline resistance and has inverted IS10L and IS10R at its end. These IS modules are oriented inwards. So this is a gene, and here at both the ends is elements are present. Here is 10, and at this end the is 10 are, that means left and right. These two is elements are present, and in between this is a tetracycline resistance gene is present. It is about 6500 base pair element having a 22 base pair, and these is element or these base pairs are always oriented in inward. Okay, they are oriented in the inward position. That means if there is A, T, C, G, then on this is element it is like G, C, T, A. Okay, so this is the Tn element. This is a diagram of the Tn element or the transposons of the Tn10 integrated into the circular DNA. This is a circular DNA and this is integrate site. Now these TTR or the transposons integrate into the plasmid here, or this is the transposons. Then these transposons move in the Tn element. These transposons then move the Tn element, and these are present in the inward orientation. And here in this case the mobilization of the host sequence, that means there is no presence of the transposons, or here the presence of the transposons, and in this case there is not a presence of the transposon. This is the mobilization of the host sequence, and hence there is a outside orientation, and in case of Tn10 the inside orientation is observed. Okay, so this is the end of our topic. Thanks for watching.